Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course Developmental Biology. I am Dr. Priya Goyal, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College of University of Delhi. In today's lecture, we will take up the topic Aging, Concepts and Theories, and we will also take up the part 1 of teratogenesis. This is the syllabus of developmental biology that we are following. First, we will start the topic aging, concepts and theories, and then we will take up teratogenesis, in which we will study teratogenic agents and their effects on embryonic development, part 1. Let us begin with the first part of this lecture, that is aging, concepts and theories. In this lecture, we will study the concept of aging, the maximum lifespan and life expectancy, various theories of aging, starting with the historical aspects, stochastic theories, in which we will study the general wear and tear theory, the oxidative damage theory, the cross-linkage theory, the mitochondrial genome damage theory, and the error theory. Then we will study the non-stochastic or programmed theories, in which we will cover the Hayflick limit theory, genetic aging programs, calorie restriction theory, chromatic modification theory, hormonal theory, and immunologic theory of aging. We'll also study the role of P53 and the insulin signaling cascade and ages in the process of aging. And the lecture will end with certain important terms like gerontology, geriatrics, Hayflick limit, and ages. So what is aging? When you see some old and some young people around you, a number of questions must come to your mind, like why does aging occur? What determines how long a person will live? Why all individuals do not live equally or age similarly? Why can some individuals run, jog, or swim so fast, or they can climb mountains so easily, while some others of the same age cannot? Is there any effect of some endogenous or some exogenous factors on the life expectancy of an individual? All these questions can be addressed if we have some fundamental understanding of the concepts and mechanisms of aging. Aging can be defined as the time-related deterioration of the physiological functions necessary for survival and fertility. Denham Herman, whose name comes quite commonly when we discuss the process of aging, he postulated that aging is the result of progressive accumulation of changes in the body which occur with passing time and which cause the increase in the probability of the disease and death of an individual. So aging is a gradual but complex and a multifactorial process where genetic, endogenous as well as the environmental factors come to play their role. The characteristics of aging as distinguished from diseases of aging, like cancer and heart disease, which come with the passage of age, affect all the individuals of a species similarly. The phenomenon of aging has two major features: the organism's lifespan, that means how long an individual can live, and second is senescence, which means the physiological deterioration which characterizes old age. So how do we define senescence? It is defined as the increase of impairment or physiological functions with age, resulting in a decreased ability to deal with a variety of stresses and an increased susceptibility to disease. When we grow old, what are the changes in the phenotype that we develop? All those phenotypic changes come under the senescence. So human senescent phenotype includes graying of hair, sagging and wrinkling of the skin, stiffness in the joints, osteoporosis which occurs as the bone calcium is lost, loss of muscle fibers and muscular strength, some memory loss, some eyesight deterioration, and also the slowing of sexual response. Here we need to understand two major terms, the first one of which is lifespan or maximum longevity, which is expressed as the maximum number of years a member of a given species has been known to survive. The lifespan or maximum longevity is characteristic of a species. For example, the humans are known to have a maximum longevity of 121 years. That means maximum any human has ever survived till the age of 121 years. In case of tortoises and lake trout, it's 150 years. Domestic dog has a maximum longevity of 20 years, a laboratory mouse 4.5 years, while a Drosophila just 3 months. On the other hand, we have the next term, life expectancy, which is defined as the average length of time a member of a species is expected to live. That is the age at which half the population still survives. Life expectancy is characteristic of populations, not of species. The life expectancy has increased considerably during recorded history of humans in most countries of the world in the 20th century, thanks to the reduced infantile mortality with discovery of novel vaccines, antibiotics, better management of infectious diseases, improved nutrition, and recent weightage given to personal and public health hygiene. Now we will study various theories of aging. To address the process of aging, till now, more than 300 theories have been laid down. However, no single theory could qualify as being the theory of aging. All these theories could be at best labeled as hypothesis or aspect theories. All these theories are overlapping as well as contradicting to each other to some extent. Metchnikoff introduced the concept that aging was caused by continuous absorption of toxins from intestinal bacteria, and he received Nobel Prize for this in the year 1908. Systematic studies that would describe the morphological, physiological, and biochemical aspects of aging began in 1950s. And the current or the modern biological theories of aging can be subdivided into two main divisions, that is the stochastic and the non-stochastic, also known as the programmed theories of aging. So, stochastic theories explain aging as chance events that occur randomly and cause cell damage that accumulate over time and cause aging, which is controlled extrinsically. Stochastic theories include the wear and tear theory, the free radical theory, the cross-linkage theory, and the error theory. On the other hand, the non-stochastic or the programmed theories view aging as certain predetermined, time-framed phenomena which is linked to biological clock or one's genetic makeup. These include the cellular aging or the Hayflick limit theory, the genetic programs, the calorie restriction theory, the chromatin modification theory, the neuroendocrine theory, and the immunological theory. So we can say in stochastic theories, aging is essentially an accident, an accumulation of damage and wear and tear to the body, which eventually accumulates over time, thus causing aging and ultimately causes death. In contrast, the programmed theories of aging view aging as an intentional process, which is controlled in a way that can be likened to other phases of life, such as puberty, which are regulated by changes in gene expression that affect the systems responsible for maintenance, repair, and defense. Thus, they consider aging to be under the control of a genetic blueprint of the organism. Let us begin with the stochastic theories first. The general wear and tear or the genetic instability theory is among the oldest hypotheses proposed to account for the human senescent phenotype. It was proposed by August Weismann in 1891, which states that as one gets older, body undergoes progressive wear and tear, while small traumas to the body build up. Point mutations increase in number, and the efficiencies of the enzymes encoded by our genes decrease. If mutations arise in the DNA synthesizing enzymes, the rate of mutations increases markedly. The faulty DNA polymerases have been documented in senescent cells. Likewise, DNA repair is important in preventing senescence. Species with more efficient DNA repair enzymes in their cells are able to live longer. Genetic defects in DNA repair enzymes can reduce premature aging syndromes in humans. Wear and tear varies on individual basis like age, stress, climate, food habits, and lifestyle. The accumulation of waste toxins, which are termed age pigments, collectively called lipofuscin, in older adult tissues, such as brain, heart, lungs, muscles, nerves, and skin, appear as age spots. These age spots deprive the tissue of oxygen and nutrient supply, and they appear little dark spots. The next theory is the oxidative damage or the free radical theory. This is the most promising theory so far. Proposed by Denham Harman in 1956, oxidative damage or the free radical theory of aging states that accumulation of free radicals derived from oxygen is the underlying reason for cell damage and alteration in cellular functions associated with aging. So what are reactive oxygen species or the ROS? They are produced endogenously when oxygen atoms taken up by the mitochondria are reduced insufficiently. The major reactive oxygen species are the superoxide ion, the hydroxyl means the free radical, and the hydrogen peroxide. So majorly these are the free radicals. The antioxidant systems in the body normally maintain low intracellular levels of these free radicals, which includes the enzymes superoxide dismutase, catalase, glutathione peroxidase, and glutathione reductase. Along with these, there are certain non-enzymatic components of the body also, like ascorbic acid, alpha-tocopherol, retinol, molecules with thiol groups such as glutathione, lipoic acid, thioredoxin, and transition metal ions such as iron, copper, zinc, and manganese, which maintain low intracellular levels of these free radicals. However, when the concentration of reactive oxygen species exceeds the antioxidant capacity of the system, as happens during the conditions of stress or unfavorable environment, the toxic effects appear as oxidative damage. The reactive oxygen species can oxidize and damage cell membranes, proteins, and nucleic acids of the body. They also promote accumulation of oxidized proteins, oxidized DNA forms, and oxidized lipids in the cells, thus leading to aging. Clinical studies have reported that reactive oxygen species are associated with many age-related degenerative diseases like atherosclerosis, cancer, trauma, stroke, breathlessness, hyperoxia, arthritis, pigmentation, dermatitis, cataract, retinopathy, hepatitis, and hypertension. A large number of experimental evidences support the free radical theory of aging. In Drosophila, expression of enzymes that destroy reactive oxygen species, particularly catalase, which degrades peroxide along with superoxide dismutase enzyme, is shown to increase the lifespan by some 30 to 40% longer than control. In the nematode C. elegans, individuals with mutations that increase the synthesis of reactive oxygen species degrading enzymes live much longer than the wild-type nematodes. Some other flies and nematodes have also shown that oxidants like vitamin E and C, beta-carotene, and glutathione, when added, act as reactive oxygen species inhibitors. Vitamin E increases the longevity of organisms when added to their diet. The next theory is the cross-linkage or glycation theory, in which we will study the role of AIDS. It was proposed by Johann Jorsden in 1942. This theory is based on a non-enzymatic reaction between reducing sugars such as glucose and proteins, lipids, or nucleic acids, which is termed as glycation. High glucose concentrations, as seen during diabetes and excess food consumption, leads to production of advanced glycated end products known as AGEs. For example, the carboxymethyl lysine or the CML, carboxyethyl lysine or the CEL, and methylglyoxylylysine dimer or the mold. These agents are responsible for causing irreversible damage to biological macromolecules, altering their structural and functional integrity, thereby causing oxidative damage to the cells. Covalent cross-linking of structural proteins in certain nucleotides damages their structures, rendering enzymes non-functional. Cross-linking of extracellular proteins, for example, the collagen, elastin, and fibronectin, stiffens them, causing skin damage, which increases with age. Cross-linking of tendons, ligaments, and muscle tissues causes loss of flexibility in the body. Thus, accumulation of ages has been linked with aging and age-related human diseases such as chronic or acute inflammation, neurodegenerative diseases like atherosclerosis, and vascular complications of diabetes mellitus. The next theory is the mitochondrial genome damage theory. This theory states that the mutation rate in mitochondria is 10 to 20 times higher than in the nuclear DNA. This mutation rate increases with age, as seen in many animals, including humans. Thus, mutations in mitochondria often lead to defects in energy production, apoptosis, and production of reactive oxygen species because of the faulty electron transport and thereby making the mitochondrial DNA more susceptible to ROS-mediated damage. This vicious circle of mutation and oxidant production eventually leads to cellular catastrophe, organ failure, and senescence. Mitochondria with mutations have a higher replication frequency than wild-type mitochondria. Thus, the mutants are able to outcompete the wild-type mitochondria and eventually dominate the cells and its progeny. The next theory is the error theory or the error catastrophe theory, which was proposed by Arjun in 1963. It is based on the idea that errors can occur in the transcription in any step of protein synthesis. Here, the symptoms include head enlargement, thin skin with age spots. If you remember, age spots are the pigment spots which are formed from the pigment lipofuscin generated in the skin, in the brain, in the liver when the person normally ages. Another symptom is resorbed bone mass, baldness, and arteriosclerosis. So all these symptoms appear, they are the symptoms of aging. And aging in these children appears even before the 12 years of age, which is their maximum age limit. A similar syndrome has also been observed in mice, which is caused by mutation in the Klotho gene. In C. elegans, there appears to be at least two genetic pathways that affect longevity and hence aging. The first longevity signal involves the insulin signaling pathway. As you note here, that after hatching, the C. elegans larva proceeds through 4 instar stages, after which it becomes an adult. However, if there are conditions of overcrowding or insufficiency of food, then this larva, instead of becoming an adult, enters a non-feeding and metabolically dormant stage, which is known as dour larva stage. The dour larva stage remains as such for up to 6 months, though an adult is able to survive only for a few weeks. So when it comes out of the dour stage, this larva will live as long as if it had never been into this stage of dour larva. Dour is a non-feeding state of diapause, a condition in which development as well as aging are suspended. A larva in the dour stage is able to show increased resistance to oxygen radicals, thereby showing resistance to cross-linking of proteins and DNA damage. However, it is interesting to note that if some of the genes involved in this pathway are mutated, the development into adult is allowed. However, the reactive oxygen defenses are still made in the larva. The resulting adults are able to live almost 2 to 4 times longer than the wild-type adults. And the pathway that regulates both dour stage larva formation and longevity in C. elegans has been identified as the insulin signaling pathway. The second pathway that influences longevity and aging in C. elegans involves the gonads. Germ cells appear to have a negative impact on longevity of the organism, while the somatic cells of the gonads act to prolong its lifespan. This diagram shows the role of insulin signaling cascade in the phenomena of aging in C. elegans. As you can see here, in the A condition, which shows the wild type, here the activation of DAF2 receptor prevents the activation of DAF16 pathway, and thus the adults are able to develop. Under starved conditions, when the food is not available, then the DAF2 receptor is not activated, and thus the DAF16 is activated. Thereby, adult development does not occur. Instead, the larva enters a tower stage in which the reactive oxygen species defenses are predominant, and this enables the formation of a long-lived doer larva instead of an adult. However, in some loss of function allele conditions, the DAF2 receptor is not activated and the DAF16 is slightly activated, which enables the formation of doer larva. And here the adult is also formed, and this adult is able to live longer than the adult which is formed under the normal conditions. And this pathway of insulin signaling is shown to be conserved among a large number of animals, as shown by the experiments done in C. elegans, mouse, and Drosophila. So this is a conserved genetic pathway that regulates aging. Here the FOXO transcription factor has been associated with longevity throughout the animal kingdom. And increase in DNA synthetic enzymes and enzymes that protect against reactive oxygen species is due to the FOXO or DAF16 transcription factor, which is inhibited by the insulin receptor DAF2. However, in case of some loss of function alleles, the adult formation is allowed, and that adult is able to live even 2 to 4 times longer. Like in Drosophila, such type of adult is known to live almost 85% longer as compared to the normal adults. And under starvation conditions, there is only and only formation of the towel larva, which is able to survive much longer, even longer than the total life of the adult in these organisms. So what are the effects of downregulation of the insulin signaling pathway? How will it affect the process of aging? Now, downregulation of insulin signaling pathway appears to influence metabolism, thereby decreasing the mitochondrial electron transport. As we have seen in the diagrams also, that when the DAF2 receptor is not active, cells increase the production of enzymes that are able to prevent oxidative damage, as well as DNA repair enzymes. And the lack of insulin signaling has been shown to decrease fertility in many organisms. The next theory is the calorie restriction theory. This theory is based on the fact that the reduction of calorie intake by almost 10 to 40% without causing malnutrition can extend the maximum lifespan in a wide range of organisms, from yeast, nematodes, flies to proteins. Calorie restriction downregulates the insulin signaling pathway, and thus it may reduce levels of insulin-like growth factor, majorly IGF-1, which is the main ligand of IGF-1 receptor, and of circulating insulin levels. Calorie restriction may slow the rate of cell division and hence reduces the chances of damage from the buildup of sugar on the proteins of the cell. Calorie restriction may limit the formation of free radicals also, thereby preventing oxidative damage to the cells. And even late onset establishment of dietary restriction has been found to be sufficient in increasing the longevity as well as in lowering the disease susceptibility in animals. However, there are not much confirmational studies in primates or even humans where scientists have seen that low calorie intake may extend their longevity, but it is not able to retard the age-associated decline of heartbeat variability or the motor coordination. Next is the chromatin modification theory, in which predominantly the certain genes play their role. The certain genes are the ones which encode histone deacetylation enzymes responsible for chromatin silencing. This is involved in the organization of global chromatin structure, which helps repair chromatin breaks. It is also involved in the epigenetic regulation of specific genes. The certain genes are present in a wide range of organisms from yeast to mammals. They are also considered longevity genes. The predominant certain genes are the SERT1 and the SERT2. Activation of certain genes is also observed in individuals which are under some calorie restriction. With aging, there is higher probability of events which involves DNA strand breakage, and certain proteins respond immediately by arriving at the damaged site in lieu of their original functions. Thus, genes that are usually silenced become active as the cells age. Alternatively, in certain parts of the body, such as brain, histone deacetylases can generate an aging phenotype. For example, in case of cognitive decline. Theory in the non-stochastic case is the hormonal or the neuroendocrine theory. This theory describes a change in hormone action with age. As we know, most of the hormone secretions are under the hypothalamus-hypophyseal axis. So with age, the hypothalamus loses its precision regulatory ability, and the downgrading of receptors which uptake individual hormones also becomes less sensitive to them, thereby causing a reduction in the secretion of many hormones. Ultimately, the hypothalamo-hypophyseal endocrine gland feedback system also gets altered. Evidence to support this hormonal or neuroendocrine theory is also given by certain researchers. With increasing age, there has been shown a decline in growth hormone secretion that results in decrease in insulin-like growth factor production in liver and certain other tissues of the body. Also, a decrease in gonadotrophin-releasing hormone from the hypothalamic neurons results in decline of luteinizing hormone, which is the primary factor in loss of reproduction cycles in female. So this makes us able to understand why the reproductive cycles are lost in females with age. The proposed reason for hypothalamic loss of regulation with age is that hypothalamus is damaged by the hormone cortisol produced from the adrenal glands. This is a gland which is located in the kidneys. Cortisol is considered to be a dark hormone which is responsible for certain psychiatric disorders or stress. Due to loss of feedback system with age, the cortisol levels are elevated. And over time, it ultimately becomes a vicious cycle of continued hypothalamic damage, leading to an ever-increasing degree of cortisol production and thus more and more hypothalamic damage, which ultimately causes aging. Next is the immunologic or autoimmune theory of aging, which states that effectiveness of the immune system peaks at puberty and gradually declines thereafter with advance in age. This decreased immune function is linked to shrinking of thymus gland to almost 15% of its capacity and altered lymphocyte functioning, decreased cell-mediated as well as humoral immune response in old age. Also, with loss of T-cell differentiation, the body's immune system can mistakenly perceive old irregular cells as foreign bodies, thereby ending up attacking and destroying its own cells. And this phenomena is known as immunosenescence. This immunosenescence has been shown to be associated with an increase in certain autoantibodies, for example, the rheumatoid factor, as well as loss of interleukins in aged people. Hence, frequency of autoimmune diseases is higher in old age. Now, with the conclusion to this lecture, let us discuss some important definitions. First is gerontology. Gerontology is the multidisciplinary study of aging concerned with physical, mental, social aspects and their effects on human body. Geriatrics is the medical science that focuses on the care and treatment of elderly which are suffering from the age-related diseases. We also studied Hayflick limit or Hayflick phenomena. It is defined as the number of times a normal cell population divides before entering the senescent phase. It was described by Leonard Hayflick and Paul Moorhead in 1961. Advanced glycation end products or the AGEs, which are also having a critical function in the phenomena of aging. They are heterogeneous compounds derived from non-enzymatic products of glucose reactions or other saccharide derivatives with proteins or lipids. These can be formed with either exogenous or endogenous mechanisms. So this completes our topic of aging. Let us now have a brief summary of what we have studied. Aging is the result of progressive accumulation of changes in the body which occur with passing time and which cause the increase in the probability of the disease and death of the individual. Senescence is the increase of impairment of physiological functions with age, resulting in a decreased ability to deal with a variety of stresses and an increased susceptibility to disease. Lifespan or maximum longevity is the maximum number of years a member of given species has been known to survive. Life expectancy is the age at which half the population still survives. That is, it is calculated as the average length of time a member of a species is expected to live. It is characteristic of populations, not of species. Biologic theories of aging can be divided into two main divisions, stochastic and non-stochastic or programmed. Stochastic theories explain aging as chance events that occur randomly and cause cell damage that accumulate over time and cause aging. This is controlled intrinsically. Wear and tear theory states that as one gets older, body undergoes progressive wear and tear and small traumas or mutations build up. Accumulation of waste products like lipofuscin in older adult tissues, which are appearable as age spots, is linked to aging. The free radical theory is based on the fact that accumulation of free radicals derived from oxygen is the underlying reason for cell damage and alteration in cellular functions associated with aging. The cross-linkage theory implies that cross-linking of biomolecules like protein and glucose during glycation to produce ages irreversibly damages the biological macromolecules, altering their structural and functional integrity, thereby causing oxidative damage to the cells. Error theory is based on the idea that errors can occur in transcription in any step of protein synthesis, which when accumulated diminish the functional ability of the original cell. The non-stochastic or programmed theories, in contrast to the stochastic ones, view aging as a certain predetermined time-framed phenomena which are linked to biological clock or one's genetic makeup. Among the non-stochastic theories, cellular aging or the Hayflick limit theory states that a cumulative effect of improper functioning of cells and eventual loss of cells in organs and tissues is responsible for aging. According to genetic program theory, longevity of an organism is primarily determined at the moment of conception and largely depends on the genes inherited from parental generation. The calorie restriction theory states that the reduction of calorie intake by 10 to 40% without causing malnutrition can extend the maximum lifespan of an organism. The chromatin modification theory postulates the role of histone deacetylases or the surgeon genes, while neuroendocrine theory describes a change in hormone action with age. And the immunologic theory of aging states that the effectiveness of the immune system peaks at puberty and gradually declines thereafter with advance in age, which is associated with higher frequency of autoimmune diseases with age. So after having a thorough study of this lecture, you can try to answer these questions. Why do we age? Comprehend your answer to the statement giving suitable evidences. How can the study of AIDS help a pharmaceutical company develop a novel anti-aging cosmetic product? How can the study of calorie restriction theory of aging help a nutritionist prepare a diet chart for persons with different physical activities? Are there any social, psychological, or economical aspects of aging? You can try to find these aspects also. Finally, can progeria be inherited? Just think over the progeria symptoms and try to give reasons for your answers, which we will take up in the next part of this lecture, that is teratogenesis part 2. These are some of the books which have been referred for preparation of content of this lecture. Hope you had a good time listening to me. Thank you. Happy learning. Now, we will start with the second part of this lecture, which is about teratogenesis. This is the syllabus of developmental biology that we are following. And in teratogenesis topic, we will be covering the part of unit 5, that is implications of developmental biology. We will study about teratogens and teratogenesis in this lecture. We will also study the principles of teratology, the effects of various teratogenic agents on embryonic development. We will learn about natural teratogens, about pharmaceutical drugs and chemicals, about endocrine disruptors, about the industrial teratogens, in which we will study predominantly the effect of heavy metals, the microbial teratogens, the recreational teratogens, the metabolic conditions in the mother, how they act as teratogens. And finally, the abnormal development that happens without mutation, which is again teratogenic for the embryo. So let us begin with part 1 of the lecture teratogenesis. What is teratogen? Teratogen is a Greek word which literally means monster formers. It is actually called teratogen because it leads to the formation of children who look like monsters, or this is how earlier they were referred to. Between 2 to 5% of human infants worldwide are born with any readily observable anatomical abnormality. These abnormalities may include some missing limbs, some missing digits, or even extra digits. Some are born with cleft palate. Some may have eyes that lack certain parts, or somebody may have a heart that lacks valves, and so forth. So teratology is the science of studying and investigating these effects, predominantly the ones which are affected because of the environment and which cause some kind of structural abnormality or defects during the embryonic development. Here we lay focus on the word environment because some birth defects are also caused by mutant genes or chromosomes. So only those disorders which are caused by environmental influence, that means they are certain kind of exogenous agents, are studied under teratology. So what is teratogen? It is any exogenous agent that causes birth defect or congenital anomalies in the form of a permanent abnormality in structure or function or restriction of growth or even death of the embryo or fetus. So teratogenesis is the process which leads to the formation of a congenital anomaly. And what are disruptions? They are the abnormalities produced due to exogenous agents, which can be certain chemicals or viruses, radiations, or even hyperthermia. Teratogens produce a consistent deviation from normal development, which can be categorized as death. It can be a malformation, it can be a growth retardation, or it can be a physiological defect. Here we have the principles of teratology. The effect of teratogen on the embryo depends on, number one, the timing and dose, or we can say the intensity of the exposure that the teratogen had on the embryo. So different agents are teratogenic in different organisms at different critical doses. Low dosage may be ineffective, while high doses may be lethal, and moderate levels cause disruptions. Now, what is critical sensitive period? The critical sensitive period of embryonic development is the period of maximum fetal susceptibility to teratogens, which is between week 3 and 8 of the embryonic period. And why this period is most susceptible? Because this is the period when organogenesis and critical differentiation occur in the embryo. Here you see this diagram shows the weeks of gestation and sensitivity of embryonic organs to teratogens. Here you see most teratogens produce their effects only during certain critical periods of development. Before week 3, as you see here, during week 1 and 2, when cleavage, implantation and gastrulation takes place, no congenital anomaly occurs. Why? Because any teratogen, if encountered at this time, will either damage most or all of the cells of an embryo, resulting in its death, or it kills only a few cells. They allow the embryo to fully recover. However, between week 3 and 38, that is the full term, when the fetus is grown to full term and is about to be delivered. So between this period, the different organs or different organ systems are affected if there is any exposure to the teratogen. And these bars show the anomalies and how they are affected by the teratogens. So here you see that central nervous system is developing throughout the gestation period, that is from 3rd to 38th week. So whole of this time it can be affected by the teratogens. So teratogenic effect is quite high on the central nervous system. Heart, as you see, starts to develop in between some 3rd week and continues till 8th week. So it is this time or the critical time during which heart will be affected if any teratogen is exposed. Upper limbs also between week 4 and 8. Eyes, as you see, from the mid of week 4 to the end of the gestation period. Lower limbs from mid week 4 to week 8. Teeth from week mid of 6 to the end. Palate in between 6 to 9. External genitalia from 7 to 38. Ear, as you see, from mid of 4 to end of 16. So here we see that central nervous system, since it is constantly forming, so it remains susceptible throughout development. The next effect of teratogen on the embryo depends on this factor, that is genetic constitution of the developing embryo. As you know, different embryos exposed to same dose of teratogen may bear different effects. That is because different alleles in the human population can influence whether a substance is benign or dangerous to the fetus. For example, the allele A2 of the gene for growth factor TGF-beta in the fetus increases the risk of embryological abnormalities from tobacco smoke absorbed through the placenta. Secondly, different alleles encoding the enzyme alcohol dehydrogenase 2 have been found to cause a result in differing abilities of the embryos to degrade ethanol. That is, when the mother is exposed to some levels of ethanol or alcohol. Now, human teratogens. They belong to different categories. Here we see the first category is of natural teratogens. So phytochemicals or ionizing radiations come under the category of natural teratogens. The second category is that of drugs and chemicals. Basically, they are all the pharmaceutical products, which include glycosides, aminopterin, antithyroid agents, atrazine, bisphenol A, commonly known as BPA, cortisone, diethylstilbestrol, commonly known as DES, and polychlorinated biphenyls, retinoic acid, mefloquine, penicillamine, and so many others. Then third category is industrial teratogens, which includes the heavy metals like lead, methyl mercury, cadmium, lithium. The fourth category is that of the microbes. Some microbes can also be teratogenic to human embryo. For example, the Coxsackie virus, the herpes simplex, rubella or the German measles, cytomegalovirus, varicella zoster, Zika virus, toxoplasma, treponema, parvovirus. So any of these, if they infect the mother, they will affect the embryo and they will be teratogenic. That is, they will cause certain kind of abnormality in the embryo. Next category is that of recreational teratogens. So these include alcohol, cocaine, lysergic acid, diethylamine, which is LSD commonly known, marijuana, tobacco. So all these things which are taken in the form of recreation by the mother when she is pregnant, when she is carrying an embryo, will severely affect the embryo. Not only when the mother is pregnant, even before the mother gets pregnant. So even then, just before conceiving, if the mother is exposed to any of these agents, it can severely affect the embryo. The sixth category is that of metabolic conditions in the mother, which includes certain diseases like autoimmune disease, which includes Rh incompatibility between the mother and the fetus, and is insulin-dependent diabetes, certain dietary deficiencies, that is when the mother is having some diet which is not balanced, which has got certain deficiencies, then malnutrition, phenylketonuria. So all these agents which affect the mother, they will affect the embryo or the fetus also, and thus they will cause certain kind of abnormalities in the fetus. So let us begin with the first category in detail, that is the natural teratogens or the phytochemicals, which we include the skunk cabbage or corn lily. The name is Veratrum californicum. So this is the photograph, as you can see here. Now this plant, it produces certain alkaloids like gervine, cyclopamine and cycloposine. And what is the effect of these alkaloid phytochemicals? They block cholesterol synthesis in the developing fetus. Thereby, they prevent the sonic hedgehog genes from functioning. And these sonic hedgehog genes play an important role in growth, differentiation, and pattern formation in the embryo. So sonic hedgehog protein, which is also required for neural differentiation, that is development of brain, spinal cord, eyes, limbs, and other body parts. So if these genes are inhibited because of the presence of these alkaloids in the embryo's body, so it will cause severe abnormalities, which includes neurological damage, absence of pituitary gland, and cyclopia, that is fusion of two eyes in the center of the forehead in the fetus of cattle or sheep. When these cattle and sheep, they graze on these plants are actually found on the highlands, that is on the mountains in the areas of North America mainly. And thus it will cause neonatal death, that is death of the child as soon as it is born. In this category also belong some other weeds like astragalus and oxytropis species. These locoweeds cause limb contracture in calves and lambs in addition to fetal deaths and abortion. Another one is quinine, which causes congenital malformations like deafness, and it may even lead to miscarriage or stillbirth. Another category in the natural teratogens is that of ionizing radiations. Among ionizing radiations, the first are X-rays. But we all know that the common diagnostic doses of X-rays are not harmful. If the level goes beyond the diagnostic doses, then it is considered harmful. Another type of ionizing radiations are gamma radiations. Gamma radiations are teratogenic at excessive exposure, and they also have a cumulative effect. The severity and extent depends on the radiation dose and the stage of cell development at the time of exposure. This radiation-induced damage to the DNA may cause chromosomal breakage and alters the DNA structure, thereby resulting in cell death, certain genetic mutations and malformations in the fetus, which causes malignancy and cell death in fetus. However, no identifiable effects in the pre-implantation period, that is before the embryo gets implanted in the uterus of the mother. The pre-implantation period of embryonic development are seen if the dose is less than 10 rads, which is also considered the threshold dose for the production of any malformations in the embryo. So that means before implantation, the mammalian embryo is insensitive to the teratogenic and growth retarding effects of the gamma radiation, but quite sensitive to its lethal effects. Now, the most critical exposure period of the gamma radiations is between 8 to 16 weeks post-fertilization. Now, if the dose is above 10 rads, then it may cause cerebral structural and functional anomalies, such as microcephaly, that is very small size of the brain and small size of the head as compared to the normal, and also mental retardation. Whereas if the dose exceeds 50 rads, then there is probability of miscarriage. Later in life also, there is seen some increased incidence of hematopoietic malignancies and leukemia. That means even if the fetus is born normal, later in life it may develop certain complications. And one rad here I have mentioned is the absorbed dose, which is delivered on exposure of soft tissue to 1 roentgen of radiation. Now, after the natural teratogens, we have the pharmaceutical teratogens, in which the first one that we'll study is thalidomide. Thalidomide is sold under the brand name Immunoprene. It was developed in 1950s by a pharmaceutical company of West Germany called Chemie. As soon as it was developed, it was widely prescribed to pregnant women as a sedative and anti-nauseant in late 1950s, as nausea is a common feature among the early pregnancy days. So it was commonly sold to those pregnant women, commonly prescribed to them. And it was also sold as an anticonvulsive drug to control anxiety and insomnia. However, it caused almost 7,000 abnormal infants to the mothers who took thalidomide in Scotland alone in the 1960s. So it was then that W. Lenz and McBride independently discovered that thalidomide was a human teratogen. And this was the time, that is 1961, that teratogens were brought to the attention of scientists and common people for the first time. So as soon as it was discovered that thalidomide has got certain teratogenic effects, it was removed from the market in November 1961. Though it was reintroduced later as anti-tumor and anti-autoimmunity drug, though its usage is restricted, that is, it is not given to the pregnant woman. Here you see this diagram shows a developmental anomaly which is caused by thalidomide. So the most noticeable anomaly of thalidomide in affected infants is phocomelia. As you see here, the long bones of the limbs of this person are reduced in length or can even be absent. These bars show that what will be the effects of having exposure to thalidomide according to the days after last menstruation during pregnancy of the mother. Absence of ear, absent thumbs, absence of arms, severe shortness of arms, dislocation of hip, malformation of ear, absence of legs, severe shortness of legs, and malformed thumbs are the several critical teratogenic effects of thalidomide. So malformed intestine, renal anomalies, heart defects, malformed intestines, absence of external ears with hearing defects, polydactyly, syndactyly, oligodactyly are among the other abnormalities caused by thalidomide. So you can infer from here how teratogenic thalidomide is. The critical period of thalidomide is between 20 to 36 days post-conception, or we can say 34 to 50 days after the last menstruation, as the pregnancy days are counted. Thalidomide affects different structures at different times of development, as is seen from this graph. The mechanism of action of thalidomide is not very clear. However, several theories postulate different things. Some theories suggest that neural crest cell migration is affected because of thalidomide. Some suggest that it causes reduced size of the ganglia and their neurons. It inhibits the cell-cell interactions, or it lowers the number of cell adhesion molecules. So these are the several postulates of the mechanism of action of thalidomide. The next pharmaceutical teratogen is retinoic acid, which we will take up in the next part of this lecture, that is teratogenesis part 2. These are some of the books which have been referred for preparation of content of this lecture. Hope you had a good time listening to me. Thank you. Happy learning.